Welcome to Opthusan. Today's topic is the embryology of the eye. In this topic, we'll discuss about the embryological origin of the eye and some clinical parts like coloboma. So let us start doing by embryology of the eye. Now, if we generally speak about the embryological origin of the embryological origin of the eye, okay, we'll get two layers of our body. The first layer is ectoderm and the second layer is mesoderm. These two layers give rise to the structures of the eye. Now if we go in a detail, so as you can see here, we can see three layers. One is neuroepithelium, the other is surface ectoderm, and another is extracellular mesenchyme. So if we go in detail, we'll find the embryological origin of the eye depends on three parts. One is neuroepithelium, the other is surface ectoderm, and the third part is extracellular mesenchyme. Now these extracellular mesenchymes are derived from two parts. One is neural crest cells and another is mesoderm. As I have said here, that is mesoderm, and these part are the other origins of the eye. Now we'll go in detail. So first point, the eye starts developing around third week of gestation. And it completes around 9th to 10th week of gestation. So it starts around third week of gestation and completes around 9th week of gestation. Okay, so next is a very important point about the genetical origin of the eye structures. A very important gene which is responsible for the development of the eye, that gene is PAX6 gene. This PAX6 gene helps in development of the eye. Now if PAX6 gene is damaged or mutated, it can also cause some conditions, like it is associated with aniridia. Okay, so now we'll go to the step-by-step development of the eye. So in step-by-step development of the eye, first we will see here, what is this? This is a layer that is called neural plate. So after forming the neural plate, here this portion of the neural plate, okay, will give rise to this kind of structure. So what happening here, there is deepening of groove. It's like a groove, and there is deepening of the groove occurs. Okay, so after deepening, it will give rise to two specific grooves, one on the right side, one on the left side. Okay, so these grooves are called optic groove. These are called optic groove. Now this optic groove, very important, first sign of eye development. Okay, so these optic groups are the first sign of eye development. Now after deepening occurs, okay, so when the folding occurs in the embryo, so this part will go like this, means this left side will go to the right side and the right side will go to the left side. Okay, and these portions will go in lateral sides. Okay, these groove parts will go into the lateral sides, and this will come in contact with this part. Okay, this will come in contact with the this part, and they will separate out. Okay, they will separate out and form two structures. That is, this part is called neuroectoderm, and this whole structure which got separated out from the neural plate layer, and which we are calling now neuroectoderm, is called optic vesicle. Okay, and this part brain part. Okay, these are the brain. Okay, now after forming the optic vesicles, there will be lateral growth. Here, see, these are the optic vesicles, and it will grow laterally like this part. Okay, after growing laterally, it will form a stalk like structure, and in the end it will form a bulb like structure. So a stalk like structure and a bulb like structure. Okay, so this stalk, we can call this stalk the optic stalk. We call this stalk the optic stalk, and bulb like structure, it is the optic vesicle, and this is the optic stalk. Now this optic stalk in future will give rise to optic nerve. Give rise to optic nerve. Okay, and this hollow bulb like structure, which is called optic vesicle, it will give rise to the eyeball and its structures. Okay, now as you can see, this optic vesicle is coming in contact with surface ectoderm now. Surface, because first this is neuroectoderm, and now it will come in contact with surface ectoderm. Okay, so this is lateral enlargement. Now this is called the surface ectoderm. Okay, now the part of the surface ectoderm which the optic vesicle is coming in contact with is called the lens placode. Very important point, lens placode. Okay, now next point. Now this, after the forming of optic vesicle and optic stalk, okay, so this lens placode will invaginate inside the optic vesicle in this manner. Invagination occur. Invagination. Okay, so this is surface ectoderm, and this part is lens placode, and it is invaginating, okay, the optic vesicle. Okay, now this lens placode, when it is invaginating, so it is forming two layers of the optic vesicle. Okay, one is inner part and the other is outer part. Okay, one is inner part, the other is outer part. This will form retinal pigment epithelium, and the inner part, it is formed by the 9 layers of retina. It will give rise to retina, and the 9 layers of retina are from the inner part, and the outer part is of retinal pigment epithelium. Okay, now as you can see, I have drawn a structure here, that is the dotted part. It is called choroidal fissure or fetal fissure. Okay, choroidal fissure or fetal fissure. Now this choroidal fissure or fetal fissure, okay, if there is a fissure in the body, it should be closed before the proper development or in proper forming a human embryo. Okay, so it should be closed off. Okay, and it closes off by 6 to 7 weeks of gestation. And if it is closed, it will give rise to a defect. Okay, we will discuss it later. Now after invagination, okay, this lens placode does a very important thing, that is pinching off lens placode. Pinching of lens placode. So after pinching of lens placode, it will form a lens vesicle. It will form a lens vesicle. Okay, now you can see here we have our lens vesicle, which will give rise to lens in later period. Now this part, okay, this is ectodermal part, and this part will give rise to the retinal layers. Okay, and there will be the supply of blood vessels, and the fibrous layer of coating will be there, and that will form eventually the whole structure of the eyeball. So if we see in a compact manner, it will be like this. Okay, so these the ectodermal part, this is the ectodermal part, and this ectodermal part will give rise to the surface structures. Okay, surface ectoderm. Now this is in green, it will form the outer fibrous layer. Now this red color structure will form inner vascular layer. So what we call the outer fibrous layer, that is scleral part. Okay, inner vascular layer, that is uveal part. Okay, and the rest of the part is retina part. I have already told you the outer part will be retinal pigment epithelium, inner part will be the rest of the 9 layers of the retina. Now this part is optic vesicle, and this is optic stalk. This optic stalk will form after closing of the choroidal or fetal fissure, that is optic nerve. Optic nerve. Okay. Now we'll see one by one which structures give rise to which parts of the eyeball. Okay. So now derivative. We'll see the derivatives of the structures. So here we can mark the layers and their derivatives. Okay. It is very important to remember these points for examination point. Okay. So first we'll see about the surface ectoderm. Surface ectoderm. As you can see, the ectoderm has two parts, that is surface ectoderm and the neuroectoderm. Okay, so we have talked about the neuroectoderm part when we are discussing about the development portion, that is this is the neuroectoderm part. Okay, and in the lateral part it will come in contact with the surface ectoderm. Okay, surface ectoderm. And this part of the brain, that is the frontal lobe. Remember, this is the frontal lobe is forming here, and the two eye are forming on the both side of the frontal lobe. Okay. So let us go to the surface ectoderm first. Okay, so this surface ectoderm give rises to the lining epithelium. Okay, it give rises to the lining epithelium of cornea and conjunctiva. Okay, first point is lining, lining epithelium of cornea and conjunctiva. Okay, and also it give rise to skin of the eyelid, okay, the skin of the eyelid, and the most important part, that is it give rise to lens placode, to lens vesicle, to lens. Okay, tarsal and meibomian glands, tarsal and meibomian glands, okay, and the lacrimal gland. Okay, lacrimal glands. Now we have completed about the surface ectoderm. These structures are derived from surface ectoderm: lining epithelium of the cornea, conjunctiva, skin of the eyelid, lens, this is the most important, tarsal and meibomian glands, and lacrimal glands. Okay, now let's go to the second layer. Okay, second layer is, we have completed the surface ectoderm. Now we'll discuss about the neuroectoderm. Okay, we'll discuss about the neuroectoderm. So first structure which is derived from the neuroectoderm is optic nerve. Okay, so you will ask, sir, why neuroectoderm give rise to optic nerve? Let us go to the picture. So as you can see, this neural plate which is neuroepithelium actually, and after pinching off we are calling it neuroectoderm. So basically it give rise to these structures. which will form later the brain frontal lobe, and on the both side that is optic vesicle, and this optic vesicle when laterally enlarged it will form the optic stalk and the bulb, and this optic stalk will give rise to the optic nerve eventually. So it is a part of neuroectoderm. Okay, it is a part of neuroectoderm. Okay, it is a part of neuroectoderm. So the first structure which neuroectoderm give rise is optic vesicle. Now second structure is retina, very important. Okay, second structure is retina, and in retina, as I have told you, it has inner 9 layer, inner 9 layer, that is neurosensory retinal part, neurosensory part, neurosensory part of the retina, and the outer layer, that is pigment epithelium, pigment epithelium. Okay, now this inner 9 layer, okay, and the outer pigment layer, it is formed from the outer layer is formed from the outer part or layer of optic vesicle, and from the inner layer of optic vesicle. I have already told you about this in the diagram. Okay, so retinal part, this is the outer part, and this part is the inner part of optic vesicle. This is optic vesicle, this is outer, and this is inner. This inner will form the 9 layers. Okay, this inner will form the 9 layers of retina, and it will form the retinal pigment epithelium. Okay, now let us go to the next structure. I have completed about the retina. Okay, and next is vitreous. I have already told about vitreous in my previous lecture. Okay, it's a jelly like structure. Next is epithelium of iris and ciliary body. Epithelium of iris, not cornea and conjunctiva, but iris and ciliary body. Okay, please pay attention, that is iris and ciliary body, not cornea and conjunctiva. Okay, next is the sphincter and dilator pupillae muscles. Sphincter and dilator pupillae muscle. Okay, so this sphincter and dilator pupillae muscles are the part of the iris. So epithelium is covered, the muscle is also covered. Okay, so this second layer, that is neuroectodermal layer, give rise to the structure, that is optic nerve. First, second is retina, next is vitreous, next is epithelium of iris and ciliary body, and sphincter and dilator pupillae. Now one very important point, this vitreous, okay, this is secondary or tertiary. Okay, secondary or tertiary vitreous, remember this thing. The vitreous which is derived from the neuroectoderm, it is secondary or tertiary vitreous. Okay, Now let's go to the next layer. Next layer is mesoderm. So we have completed about the surface ectoderm and the neuroectoderm, and we'll go to the mesoderm. Okay, so we can say that is we are doing the layer of mesenchyme. It is derived from mesoderm and neural crest cells. So first we'll see about the mesoderm origins. Okay, so mesoderm gave rise to a very important muscular structure that we called extraocular muscle. Extraocular muscle. So how many extraocular muscles are there? If we say directly, some will tell 6, some will tell 7. So which is the right answer? The right answer is 7, not 6. Okay, because the muscles are superior rectus, inferior rectus, medial rectus, lateral rectus, superior oblique, inferior oblique. So these completes the 6 muscles. Okay, but there is another muscle, that is levator palpebrae superioris, very important. Okay, very important. So these are the 7 extraocular muscles which are derived from the mesoderm. Okay, now next, which structures are derived from mesoderm? Now the other parts are derived from the mesoderm are these structures. I'm making a list. I'll fill it one by one. Okay, so first structure is choroid. Okay, temporal part of the sclera. Part of sclera. Okay, and then vascular endotheliums. Okay, now there is some changes can happen here also for your examination purpose. So we have a layer that we are calling neural crest cell layer. Okay, so if we can see the structures which I have studied until now, that is lining epithelium, let us do the marking. 1 2 3 4 5. Okay, 6 7 8 9 10 11 structures. Okay, and some other structures like ciliary muscles, stroma of cornea. Okay, ciliary muscles, ciliary muscles are coming from neural crest layer, stroma of cornea. Okay, choroid also, etc. These are coming from neural crest layer cells. Okay, so if we can go in detail of the neural crest layer cells, okay, it have two part. One is mesenchymal part, and the other is neural part. Okay, so these two layers will give rise to different type of structures. So this mesenchymal part, okay, will give rise to bony part of the orbit. Connective part of the orbit. Okay, so bony part and connective part, so it give rise to orbit, we can say directly. Next is trabecular meshwork. Okay, trabecular meshwork. Okay, I have already told about the ciliary muscles. Okay, and now just now I have told you about the mesodermal origin of the temporal part of the sclera. So the temporal part of the sclera is originated from the mesoderm. Okay, and the neural crest layer cells give rise to the sclera part except the temporal part, very important question. Okay, now the other structures are corneal stroma and corneal endothelium. Okay, so these structures are derived from the mesenchymal part, that is bony part of the orbit, connective part of the orbit. So that means the orbit, trabecular meshwork, ciliary muscles, scleral part except the temporal part which is derived from mesoderm. Okay, mesoderm, and the corneal stroma and corneal endothelium. Now this neural part of the neural crest cell layer give rise to Schwann cells and melanocytes. Schwann cell and melanocytes. So we have completed about the derivatives from each layer. Okay, so first we have done about the surface ectoderm, next we have done about the neuroectoderm, and then we have done the mesenchymal part, that is mesoderm and neural crest cell layer. Okay, so we'll go to a clinical part now, that is called coloboma, that is called coloboma. Now what is this coloboma? Now if we can see, this coloboma has a Greek origin. Okay, Greek origin of name, that is Koloboma. This is Greek name, that is coloboma. It means missing part. Coloboma means missing part. Okay, so something is missing in the eye. Now I have already told you about the choroidal or fetal fissure. Okay, so it is a congenital malformation. It is a congenital malformation which occurs, okay, which occurs due to non-closure, due to non-closure of choroidal, due to non-closure of choroidal or fetal fissure by 6th to 7th week of gestation. Okay, non-closure of choroidal or fetal fissure by 6th and 7th week of gestation. Okay, so there are some types of coloboma. We will discuss it one by one. The first type of coloboma is eyelid coloboma. Okay, first is eyelid coloboma. So if we draw a diagram here, so nose, and this is the eyelid normal. If there is a coloboma, there will be a defect in the eyelid. Okay, so eyelid has three parts. If I can divide it in like this, that is the lateral part, the middle part, and the medial part. Okay, so this eyelid coloboma is common in the upper eyelid, not in the lower eyelid. So this is upper eyelid. Okay, this is upper eyelid, most commonly seen in the upper eyelid and in the junction between middle and medial 1/3. Okay, middle and medial 1/3. So the eyelid coloboma is most commonly seen on upper eyelid between the junction of the middle and medial 1/3. Okay, middle and medial 1/3. Now due to this the eyelid looks like, so this is our nose and the eyelid look like this. So there is a defect here, so there is a defect. Okay, so due to this defect there will be some complications, obviously. Okay, so what are the complication? What is the most common complication? We can say most common complication, because due to this defect, when the patient will close eye, this part will still be exposed. Okay, this part will still be exposed, so the sclera will be exposed direct to the sunlight more continuously than the normal person. So and the cornea also, so then it will cause the exposure keratopathy. It will cause exposure keratopathy due to excessive exposure to the sunlight. Okay, or light. It will cause irritation to the corneal part and it will cause exposure keratopathy. So what can be the treatment remedies? We can do lubrication by lubricants if the defect is small. Okay, if the defect is around moderate in size, we can do patching. But if it is a very large size of defect, it should be handled by surgical closure. Okay, after surgically closing this part, the shape will be like this in normal. Okay, so this is about the eyelid coloboma. Next type of coloboma is, that is iris coloboma. we have completed about the eyelid. Next is iris coloboma. Now this iris coloboma has two types. Iris coloboma has two types. That is, one is typical. If one is typical, the other will always be atypical. Okay, so what is the difference between typical and atypical? If the coloboma is seen in infranasal area of the eye, then it is iris coloboma. Okay, infranasal part of the iris, then it is infranasal iris coloboma, that is typical. And other than infranasal, anywhere, it will be called atypical iris coloboma. Okay, atypical iris coloboma. Now if we draw a diagram here, this is a normal eye. Okay, this is iris, this is the pupil, this is sclera, this is sclera, cornea is covering. Okay, this is iris and this is pupil. But if there is infranasal defect, it will look like this. Cornea will remain normal, but it will look like this. Okay, the defect will look like this. So there is a defect in infranasal area of the eye. So this type of defect is called iris typical iris coloboma. Okay, typical iris coloboma. Now this iris coloboma can be associated with, it is not a solo disease, it will always be associated with ciliary body or choroid or retina or optic nerve colobomas. Okay, now it is usually bilateral. It is usually bilateral. Okay, now it may lead to, okay, it may lead to nystagmus first. Second can be glaucoma, and another can be squint. Okay, this coloboma, iris coloboma, may lead to nystagmus, glaucoma and squint. So what are the clinical features here? First is diplopia. First and foremost is diplopia. Now as you can see here in this picture, okay, so if we remove this portion, now here will be the lens. Okay, lens will be present here. Okay, so lens will be like this. In the inside part I am drawing just to show you, the lens will be inside and this part will be can see from the outside. Okay, all right. So this lens is dividing this part of the iris into two part. Okay, dividing this part of the iris to two part, that is the first part and the second part. Okay, now in first part, when light will go, okay, it will encounter the lens which has plus 16 diopter of power. Okay, and when light will pass through the second part, it will go, but it will not encounter the lens and it will go through 0 diopter power. Now there is always a power difference between these two parts. Okay, and when light are passing, due to 16 diopter and 0 diopter, there is a difference of plus 16 diopter. It will cause, okay, light to be changed in direction. So it will give rise to two different type of pictures on the retina. So it will cause diplopia. Okay, one part is going through the lens, that is by 16 diopter, and one is go through this part, that is has 0 diopter power. Okay, so two type of images will be found on the retina and this will cause diplopia. Okay, now second is photophobia. Okay, and the third is cosmesis. Okay, first is diplopia, second is photophobia, third is cosmesis. Now what are the treatment? So for this type of coloboma, the treatment is cosmetic contact lenses, or surgical closure by suturing the defect. Surgical closure by suturing the defect. Okay, the other type of colobomas are retinochoroidal coloboma, retinochoroidal coloboma, and other is lens coloboma. Now this lens coloboma is not a true coloboma, not a true coloboma. Lens coloboma is not a true coloboma because it doesn't follow the course where the choroidal or fetal fissure fissure is not closed around 6 to 7 week of gestation. Okay, so it means that this lens coloboma occurs, but it doesn't due to choroidal or fetal fissure not closing in the 6th to 7th week of gestation. Okay, so by this we have completed our lecture on embryology. Okay, next we'll discuss about the layers of the eye and furthermore. Thank you.